आ गए हैं अभी, वही वो सामान समार हो गए थे। हाँ रिकॉर्डिंग ऑन है। हो हाँ ठीक चल रहा है, नहीं कोई दिक्कत नहीं, लोड ले ले रहा है, थोड़ा सा क्वालिटी कम है। ठीक है। To an enlightening session that uses epigenetic and ontology in ways that promise hope for innovative cancer therapy. Thank you. ठीक है अच्छा अच्छा, shall we start? It is my honor that Dr. Santosh invite me to talk about epigenetics on cancer. So I'm working on the cancer, I think from last 10 years. But few of the paper published on the epigenetics and remaining are only on the pure immunology. Okay. So can you slide full page पे? अच्छा अरे भाई, okay. So I think from last 1 week you people studied about epigenetics, its basics, right? So everything is now clear. So I will not talk much about epigenetic modification, its basics, what are the factors. So we will just remind previous stuff what Professor क्या नाम है? Kunal Rai. Yeah. I met him also in MD Anderson because Baylor College of Medicine and MD Anderson is just side by side. So cancer as an epigenetics, let's begin its by history. How many people know him? Have you read any time? No. So like he is the first scientist who discovered basically, but not recognized during that time. People thought that this is impossible. Like in the current scenario, we know that we can talk by mobile without any wire. But 20 or 30 years back, people don't believe that we can talk like this. And we can visually talk as well. So same thing was happened with him as well. But really he invented in 1920, and first time the term is given by the Waddington Quine, the epigenetics. So I hope epigenetic modifications, now you people understand what is the DNA, histone, everything. Other than that, what are the things can also regulate the transcription of gene? Can anyone tell? There are several factors. Okay. Huh.
Transcription factors affect the transcription, then the phosphorylation, methylation at different sites, acetylation on the DNA.
They are all histone modification. Other than that,
we have enhancers, super enhancers,
yes,
promoters. These elements also affect the rate of transcription.
Yeah. So Kunal sir also teach those things. Wow, good. But few of things I will also cover, because lines, signs, you know, and enhancers, super enhancers, suppressor, they all play role in the gene regulation, gene expression. And all this disease is appearing because of sometime over suppression or over expression, uncontrolled expression. Okay. So what basically is the cancer? No, just terminology of the cancer. Uncontrolled cell growth. That is the cancer. There is a no control. They don't know how to stop. So they are continuous growing and making the cells. But in our body, everything has switch on, switch off stuffs, right? When you eat any food, your digestive enzyme start to secrete. They are not secreting all the time. And to start, there is a trigger. To stop, there is a trigger. Okay. So like in our body, several dangerous stuff is also made, like HCl, where stomach, its pH is how much? Can you hold on hand HCl? No, but our stomach holds. So every day our intestine get renewed after every digestion. So that's why if you make the routine for your food habit and that is getting changed, people get acidity or problem in digestion. So our body cycle always understand when we are going to eat the lunch, when we are going to eat the dinner. So during that time you feel the hungry. It is not like that you feel any time. So that particular time usually you feel, right? Because your machinery calculate everything. Like these days we are reading the using the AI, but our body is itself is AI. It's know how, your factors, your understandings, and even your behavior. So it prepare everything accordingly. So even though in 10 individuals, they have all different, their thinking, their stuff. So their body structure is also changed accordingly. Okay, let's begin then. So short summary is like introduction to epigenetics in cancer. Genetically alteration in cancer was normal cells, methylation, histone modification, full epigenomes. Okay, here one terminology came, epigenome. What is epigenome? Huh? On the genome. More elaborate, anyone? Epigenetic, these are two different terminology. Yeah.
Or epigenetics are factors which are not in the sequence of the DNA, but they're added onto the DNA, which change the expression of the same.
Someone more about epigenome? It is getting closer. Okay. Okay. See, there is a one major difference is that epigenetic we are talking, epigenome is we are talking home full epigenetics. So that makes one holes in the cheese alignment. That is the system is happening. So if that all genetic modifications clustered together at a certain, then it is the initiation of the disease development. It is not only cancer, it can be any neurological disorder, it can be any autoimmune diseases as well. So there are several diseases we don't know the etiology, how they starts. Like some autoimmune diseases like on the skin, and talk little bit about IBD on the in the last lecture. Okay, and examples and what we are doing right now, that will also I will talk. So but before that we will understand the basics, how they starts. And so can we use the epigenetic understanding or epigenetic machinery for the therapeutic aspects? Yes, because epigenetic is in DNA sequences. It is just modification on the expression of particular genes. We are not inserting anything, but everything is doing by the secretary, right? Same thing is happen. Secretary will tell that today we have to travel there. So by analyzing the epigenetic markers, we can identify the cancer cell markers, tumor prognosis, chemotherapeutic sensitivity, some future ideas in cancer epigenome as well. Okay, this you people already read. So like full 10 on, so maybe possible there is a overlapping stuff. So like you can see our lifestyle, our food, diet, aging, environment, all they can make the change of genetic alterations. Okay, that is creating the genome instability. That is the main reason is normal cell can convert into the cancerous cell. many other diseases also linked with the epigenetics. I already told you people, but now my is teaching on the cancer, okay? Because I'm working on the three aspects. So transcriptional addiction in the cancer. ठीक है, okay. Addiction जैसे in normal, see our gene regulation, everything is you can find the example in your daily life as well. Addiction is same, okay. So cancer that invariably leads transcriptional programs, these dysregulate cancer cells to become इसी बारे में, okay. So in English, okay, because sometimes, okay. So so transcriptional control, if this, can you read the 4 line? By genetic alteration in cancer, by transcriptional dependent can develop an consequence of the dysregulated programs, and how these dependencies provide opportunity for novel therapeutic. Understand all these mechanisms to find out the therapies. So going on, plasticity, elastic and plastic. You people understand? Okay, good. I have good audience. Epigenetic plasticity and of the cancer. So I will understand this slide. So next, this figure also you people understand properly, okay? Environment factor on gene expression in DNA. The molecules is called the methylation turned on. Methylation turned off, genes on. Acetylation is happening on DNA or both? Both, है ना? Methylation, both. If acetylation is increased, what is going to increase? Can it reduce also? Huh? Okay. Okay. So it is a universal in methylation? Yeah. Okay. Good. It is very, very everything. Meiotically and mitotically and meiotically. This is just simple. Mitosis and mitosis is happening in which type of cells? And meiosis? Okay. So if I say mutation or like epigenetic modification happens in the mitosis, active cells, can it pass to the next generation meiotically? Okay. Any example of those diseases in both cases? In cancer, skin cancer, anything, okay. ये थोड़ा डिफिकल्ट है ना, जर्म लैंग्वेज होना. अच्छा, ओके. तो CRC, hemophilia can be possible. Also sometimes it goes other than cancer. You people can, and a few cases found in case of leukemia also it's prone. CRC happening in which organ? What is CRC? Colon, colorectal carcinoma. So usually that's why one study is going on the CRC as well. Around 10% patient of the IB to the CRC, especially ulcerative colitis, because that is affecting the colon portion. Okay, and one new study is going on, fibrosis in the colon. What is the fibrosis? फाइब्रोसिस और नेक्रोसिस में क्या अंतर है कुछ? हाँ, फाइब्रोसिस में. Okay, okay, you people know something. Okay, so this is also linked with epigenetic modification changes. Okay, And like molecule modification in chromatin is DNA methylation, you people know. Modification on H3H4, acetylation, phosphorylation, methylation. So acetylation and phosphorylation is happening only on histone or other proteins also. This comes in the post-transcriptional, post-translational modification. So in attached methylation, methyl, phosphorylation, this is also kind of post-translational modification. In case of enzymes or signaling cascade, ठीक है ना? So sometime on or switch off is dependent on the phosphorylation. So like sir already, if he shows the gel picture, it's difficult to understand, right? But phosphorylation usually people identify the protein gel, है ना? Okay. And here you can see ncRNAs, non-coding RNA, microRNA and comes in that domain. Chromatin remodels and SNP. So it is microRNA and siRNA in quite hot field into the recent. It is going on from last 10 years to 15 years because people that they also play role into the gene expression regulation. Okay. तो ये तो अभी पता ही होगा, पहले ट्रांसक्रिप्शन होता है फिर ट्रांसलेशन होता है, ठीक है. हैपनिंग एट द पॉइंट ऑफ and after discuss about post translational modifications. Okay, this figure already sir shows you. Yes, there are not many figures, only this kind of. So heterochromatin. So I think you people studied already in high school in inter. Okay. So you for any gene expression, euchromatin structure is needed. In heterochromatin, they cannot. Okay. Have you people already know that methylation, hypermethylation, and you can see here in between NCRNA, non-coding RNAs. So like in this way, make the triplet of the, usually DNA is duplex, right? So they can stop the gene expression by this as well. So ncRNA is can be linear as well, can be, it is not necessary they are. So table linear or circular? Confusion है सब में sir. Okay. Okay. Epigenetic markers in the normal and cancer cells. Marks. So the cell type, cell and tissue type. So our tissue type like your liver cancer, skin cancer, breast cancer or brain cancer, all they are different type of tissues. Regulation is also changes according. So DNA methylation, CpG isolate, transcription starting sites, UTR's area, high, this already you people read. X inactivation, hypermethylation, repeat transposable element and hypermethylation. Application में acetylation, methylation, phosphorylation, smolylation. Ubiquitination, where it's bind in case of epigenetic. If we, okay, and what is the role of this? Okay, and what is ubiquitination? So sometime like when cell try to that time usually degrade, because during that time it's need to be the actively duplication of the chromosome, every chromosome, right? Okay. So during that to be full-fledged replication of whole chromosome, every, okay. ठीक है. So but they are also playing role in case of gene expression. So like few our body in our cells, there are, if they switch off, like for what type of processes needed, always gene should be active. Huh? Production of enzyme, you can say metabolism is always going on, like glycolysis, TC, actin filament, ठीक है, ER, mitochondria. Okay, one thing came to my mind. Does mitochondria has its self genome? If we talk about how evolution make the mitochondria in eukaryotes, which type of bacteria? Aerobic or anaerobic? Anaerobic? Aerobic. Okay, chalo, ठीक. Same thing happens in case of plant cells. There is a, other than mitochondria, there is a one more machinery, chloroplast. What type of bacteria? Bacteria. Okay. Is also kind of, so इसका sumo complex बनता है, ठीक है ना? So every word has name, I don't remember. S U means like sumo, U means ubiquitination. So ubiquitination like protein. Okay. So until now we was talking about epigenetic modification. Now here you can say that just now, sir, and super enhancing to enhancer, how it, like few example is already I can tell you, here is given the some enhancer beta WNT, they are transcription factor, they go and bind on the enhancer sites, basically is the DNA. Okay, so like way a particular gene's expression we can increase several fold, but in some cases like in immune we always try some cytokine production is tremendously around 100-fold. How it happens? There is another of regulation in our body is copy. Few genes has several copy number. Copy number increase 1 to 5, and one gene expression is started from 1 to 10-fold. Then total increased 50-fold with the 5 copies. So in few gene cases we have the our body also regulate by the copy number as well. Okay, so you can also see few people are super active. Why? It may be possible from their office numbers, so pass on in the generation as well. Okay, so all the enhancer and where in which scenario they occur. So enhancer, one transcription on one enhancer site rating. If multiple transcription factor align together and they are controlling the gene expression, then it's become super as well. And enhancer sites may be possible, not only one, may be possible there's a 3, 4. Sometimes they make the loop also. Maybe sir already teach those things, right? Okay. 3 transcription factor you can add 3 TCF3. When they are at the same places and even leaf TGF beta WN, they pull on the same site, then they acting as the super enhancer. And if they are individually, then they just call the enhancer. इस स्लाइड में और कुछ है क्यों नहीं? Okay, so try to understand how a normal cell, so can make the primed, yeah, again epigenetic change, can lead to move cancerous side, but not real. If there is a DNA alteration and these two coupled together, like in this case, you can say cancer initiated. So because of that, like single factor usually not governing towards the development of a disease, multiple factor aligns. So that is also one type of control in our body. It is not just dragging by one factor, otherwise everyone will get the disease. So all is turning to the cancer progression. So, but you can see usually cancer starts from one cell. I think in second class I will teach you what is the difference between cells and there is a some special cells. There is a terminology called the stem cells. Have you people read this terminology? Okay, so in detailed mechanism we will go in second class. But for the time being, we are going to understand epigenetic and cancer. Okay. I think still many slides remain. Okay, epigenetic elements like there came differential DNA repair, hot spots in active promoters. So you people have already the basic understanding of the this when is accumulated for a particular oncogene on the site or enhancer site or suppressor site, they lead to the disruption. Okay, but not only one place, multiple gene if affected, then in that case cell is going to cancer develop. C binding sites are frequently mutated in cancers, disruption of chromatin folding domains. So more words, chromatin folding. Chromatin पता है? हाँ. So if fold, when cell is dividing, folding is not proper, also they can be become cancerous. Okay, folding domains by somatic genomic rearrangement in human cancers. So we have very fantastic mechanism into the cells to stop to development of the cancer, like repair mechanism. If cell some, then cell is undergoing for the apoptosis. है ना, best word is if there is any protein something is happen, then where they go for degradation. So don't body is making only 1 or 100 proteins. I mean at every second there is a thousand protein is, but most of the protein is undergoing for the degradation if they are not properly folded. है ना, आप कोई भी चीज बनाते हो हमेशा सेम क्लोन नहीं बनती. कंप्यूटर इज नहीं इज ओनली इन कंप्यूटर इट इज पॉसिबल टू कॉपी एंड पेस्ट. Otherwise if you are making anything by mechanical possible replicate. Checkpoints is there, these accumulation of this kind of aberrant processes. Okay, some super enhancer, there is a kind of DNA break can increase the oncogene and DNA break near to the enhancer, insertion of the MTF binding sites between two enhancers. And here you can see that normal gene case they make the loop and enhancer can, but in the loop is not properly made, some mutated anchors, so they can the kind of super enhancer or enhancers for mixed gene regulation. You enhancer can be present either side of the positive side, negative side also. And okay, before that moving forward, have you people think that bacteria also have the epigenetic modification? This we all talking about always for the years. क्रोमोसोम होता है तो क्या होता है, देखो हिस्टोन मॉडिफिकेशन और फोल्डिंग एवरीथिंग राइट विदाउट प्रॉपर हमने heterochromatin is very important. Chromosome, if there is no chromosome in bacteria, epigenetic modification is possible or not? तो उसमें कैसे कंट्रोल होता है? Anyone? Any idea? Okay. Just think that if there is a no histone, so only histone is not making the, other than histone is also, what are more clues? हाँ, DNA methylation, acetyl can be possible. फास्फोर है तो हिस्टोन में होता है ना, अभी तो पढ़ा है पोस्ट ट्रांसलेशनल मॉडिफिकेशन. ठीक. So those kind of mechanism is present also in bacteria, role a gene expression. Okay. There is a basic one more differences is there. Bacterial chromosome usually have the auric site, but in our cases is also clustered. We are having the in particular introns and introns. Those introns come enhancer as well or suppressor. But in bacteria there is a no intron. ठीक है. And in bacteria one more difference, big differences. In being, if we have 5 kb gene, 5 kb DNA sequences, kilobase, and if I give you one condition, 1 kilobase pair, 5 in bacteria, I give same 5 kb total DNA, each gene will having 1 kb. It can be possible less or more. Why? Yeah, because bacteria always have the several overlapping genes. It does not have the intron as well. So that 10 kb and there are 1 kb gene, but from one first gene, in middle of the first gene also second gene can be started. So like this there is a one controlling site in bacteria can control, because bacteria organism, it is kind of in, right? So it makes it full. जैसे ट्रेन एक ही इंजन होता है ना, सेवरल डिब्बे हो सकते हैं, लेकिन जब एरोप्लेन चलता है तो उसमें सब कंबाइन है. So